Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself. So thank you for joining us today. It is now my pleasure to introduce our speaker from Mirus Bio.
Great, thanks so much, Leah. And thanks to everyone who's joined us today to listen to this presentation on efficient and scalable GMP manufacture of AAV and LV, as well as those that are considering the TransIT VirusGEN reagents and enhancers for their own specific and unique workflow. During this presentation, I do want to briefly introduce you to Mirus Bio and Gamma Bio, our sister company, as well as cell and gene therapy, the promises that these therapeutics hold for curing diseases, as well as the challenges that we as developers face in meeting those demands. We'll talk about transfection optimization and scaling considerations for both recombinant AAV and LV manufacture, as well as the workflow flexibility and cost savings that the TransIT VirusGEN reagent can offer you. So we'll go through those details as well, followed by the Q&A session. So please do, as we had mentioned, write your questions in the chat box, and we will address as many as we can following the presentation. So a little bit about Mirus. One thing that has always impressed me about Mirus Bio is that this company really is truly as dedicated to its science as we are to the scientists that we support with our products. Mirus was founded in 1995 by 3 researchers out of the University of Wisconsin-Madison, really truly pioneers in gene therapy, even in that day, with an in vitro and an in vivo component. And of course, we focused a little bit more heavily on the in vitro side of things, developing novel lipid and polymer-based transfection formulations, which are designed to both improve your transfection efficiency, as well as reduce cytotoxicity that you would often see with liposomal formulations, as well as some PEI-based formulations. And of course, we're committed to finding optimal transfection solutions, so reagents as well as strategies for many different cell biology applications, including virus production, stable cell line generation, CRISPR-Cas gene editing, protein production, stem cell biology, and for those working in really difficult to transfect cells like T cells and B cells, we also offer electroporation strategies and solutions to enable you to do that as well. And last but not least, we have a TransIT mRNA transfection reagent that's been heavily used in the development of these modern mRNA-based vaccines as well. So really solutions to support the gamut of cell biology applications. And of course, there is our technical support team as well. So when you evaluate a Mirus reagent and you have questions, you're able to access our tech support team, which is composed of our R&D and application scientists, so people that have worked in the lab with the reagents that you're learning about and working with. So when you contact us, you'll talk to a scientist rather than a call tree. And we want to make sure that you feel supported as you explore these processes and these reagents. And of course, just this last year, Mirus has partnered with Gamma Biosciences and joins other Gamma Bio companies, such as Astrea Bioseparations, Univercells Technologies, Nanoparel, Biomagnetic Solutions, and Mirus, to enable innovation and provide end-to-end solutions for cell and gene therapies, vaccines, and biologics. So ultimately, the goal for the Gamma Bio family is to enable biopharmaceutical innovators and manufacturers to both develop and deliver complex therapies to patients more quickly and efficiently. All right, so now into the cell and gene therapy section, where we'll address both the promises and the challenges that this field is currently facing. So cell and gene therapies are unique in that they utilize viral vectors like lentivirus and adeno-associated virus, or AAV, as vehicles for direct delivery of therapeutic genes to patient cells, whether that's in vitro or in vivo. For gene therapy, the transgene is packaged within the virus itself, and then that virus is administered to the patient as the therapeutic. And conditions that have been treated with AAV-based gene therapies would include spinal muscular atrophy, inherited retinal diseases, and of course there are new indications being studied such as hemophilia and Parkinson's as well. And currently, there are 2 FDA-approved gene therapies that utilize AAV, and those are Luxturna, developed by Spark for inherited retinal diseases, and Zolgensma, developed by Novartis for SMA. Cell therapy is a little bit different in that engineered cells are the therapeutic, and in this case, patient cells are extracted from the body, and then they are manipulated with the lentivirus vector to express the transgene that is going to really enable those cells to be re-administered to the body and attack cells such as cancer that are obviously not there to improve patient health. So conditions that have been treated with cell therapies using lentivirus would include lymphoma, leukemia, but also more recent studies involve looking at diseases like HIV and beta thalassemia. There are currently 4 FDA-approved cell therapies utilizing lentivirus, and those include Kymriah, Yescarta, Tecartus, and Breyanzi, all of which are designed to treat different types of leukemias and lymphomas. And of course, I think we're all aware that cell and gene therapies are on the rise. Just 5-6 years ago, there were only a few out there being studied and looked at, but then at this point, we have over 1,000 cell and gene therapies worldwide, or therapy developers worldwide, I should say, as well as over 1,200 ongoing regenerative medicine and advanced therapy trials worldwide. So there's a lot going on, and the predictions are only looking higher. So there's going to be more and more of these therapies being developed as time goes on. And so, of course, for us, looking at the workflow of this, how do we support this? You can kind of break this down into 2 waves. So the first wave really met the demand of those initial studies, so the development of Luxturna and Zolgensma, where adherent cell culture was sufficient to, in terms of scalability and production levels, to support the process yields that were required. Of course, this was very high cost, which was quite tolerated because it's an emerging modality. There's obviously immense potential for both the patient as well as just the field. However, that isn't necessarily sustainable, right? Your number of assets was small, the average dose wasn't huge, the patient population for each indication was also pretty small. But in this next phase, of course, with the promise that we've seen in cell and gene therapy so far, the ability to truly cure diseases, there is an immense demand. So there's 100 to 1,000-fold greater demand. The number of assets has increased significantly. The patient dose is also higher. And we're also looking at indications that address greater patient populations. So, not in the 1 to 20,000 bucket anymore, but 10 to 100,000 patients. And for this, we really need greater capacity. So the shift has been toward suspension cell culture instead of adherent because it's much more scalable. We need higher yields. It's very, very clear that we need processes that give you higher titers per mil of culture. And of course, the cost of these also has to decrease to enable economical manufacturing of those high-dose assets. And that really is what drove us to develop the TransIT VirusGEN transfection reagent at Mirus Bio. So VirusGEN was developed by screening our immense libraries of lipids and polymers to find that exact perfect synergistic formulation that enabled both production of lentivirus and AAV that worked better than the reagents that were on the market, and was also enabled cell and platform flexibility for our researchers. We have people working in adherent formats, suspension formats, different types of hex cells, different medias. We wanted a formulation that was compatible with all of that so that we could support as many teams as possible. VirusGEN is available in research grade, select grade, and GMP grades, which means it's really designed to support you from the beginning to the manufacturing process of these therapies. And of course, last but certainly not least, VirusGEN should be worry-free for anyone using it, and that there's no commercial license required, so no one's going to come and ask you to pay an immense fee for just using this reagent. That is not a thing that Mirus does. It's also chemically defined and animal origin-free. And in addition to the reagent, we've developed kits, VirusGEN AAV and LV kits that contain both that transfection reagent as well as application-specific enhancer formulations for AAV and LV. And these enhancers most often will boost your functional titers by 2 to 10-fold over a TransIT VirusGEN reagent alone. So definitely worth testing and evaluating in your system. And these also do come in RUO and GMP configurations. And this takes us to our first of 3 polling questions for you, the audience, to address. And so this one is a select all that apply, meaning we want you to choose anything that applies to your current situation. So the question is, what are your biggest hurdles when developing a large-scale viral vector production platform? Is it scalability and expression variability? Is it cost of materials and labor? Inadequate viral vector expression levels? Meeting accelerated timelines? Regulatory requirements? Or there's also the NA option for those that are not currently developing a therapeutic. So please go ahead and select your choices, and we'll give you a few seconds before moving into the next section. All right, so in this next section, we want to really kind of get into the weeds of optimization. What parameters are critical to enable the highest possible LV and AAV generation within your specific workflow? And of course, one of the key parameters that you're very likely going to look at would be transfection reagents. So there are many reagents on the market. There are a smaller number of reagents that are GMP compatible. And here we're looking at the TransIT VirusGEN Reagent or kit. So on the left-hand side, we've got AAV production. The right-hand side, LV production. The gray bars with the red outline is VirusGEN alone. The red bars is VirusGEN plus the enhancer. And then we're looking at comparisons to 25 kilodalton PI, as well as a competing GMP reagent. It's also PI based. And one thing I want to point out about most of the figures that we're going to share with you today are that we're looking at functional titers. So if you look at the axis here on the left, functional titer means we're measuring only the viral particles that are capable of producing transgene expression in a cell type, in a target cell. So functional viral particles. So you may be used to seeing genome copy titers, which would be higher. So E to the 9th, E to the 10th, E to the 11th. Functional titers, of course, are going to be a little bit lower. So E to the 7th and 8th are very common here. So just to explain what we're looking at in terms of titer, the other point that I want to point out in each of these figures is the cell type and the media that we're using for expression, because we've tested quite a bit. And as I mentioned before, we want to enable that flexibility. So you can use your cell type or the media that you prefer, but we'll also give you an idea of what we have seen be beneficial in our own workflows here. So here we're using Freestyle 293F cells, ground in Freestyle F17 media. And again, looking at AAV production and LV production, clearly just the VirusGEN reagent alone will enable a good 3-fold higher titers than the competing reagents. But when you add in the enhancer, we see about a 2-fold further increase in functional titers over reagent alone. So definitely worth checking out different reagents for your workflow. And another parameter that can be overlooked, but is definitely critical for both adherent and suspension cell cultures, is cell density. Here we're looking at LV production in adherent cells, so HEC293T17 cells grown in just the standard DMEM plus 10% FBS. Again, these gray bars are reagent alone. Red bars are that additional boost that you see from the enhancer. And so it's really clear that when you're seeding too light, when your cells are 60% or thinner in terms of seeding density, not so great for virus production. We definitely want cells to be in the 80% range for both AAV and LV. For LV, you can even go a little bit higher, and that can definitely help as well. So 80 to 90% for lentivirus production. Definitely something to keep in mind. If you plate cells and they're just not thin enough, or perhaps they're too confluent, it's probably worth reseeding if you can to make sure that you're in that 80 to 90% confluent range. Cell density is, of course, also important in suspension cells. And here we're looking at AAV production and LV production. In the top graph, we used XB293F cells grown in XB293 expression media for AAV. And again, if we look at just the gray bars, we see that max production is right around 2 million cells per mil. But if we start utilizing the enhancer, we do see that if you bump up your cell density just a little bit, so moving somewhere between 2 and 3 million cells per mil, we do tend to see a little bit of an increase in production and performance here. So it's definitely worth testing a few different densities between probably 2 and 3 million cells per mil to make sure that you found what's optimal for your specific serotype, your cells, and your media. Lentivirus is, I guess, even more straightforward. So without the enhancer, 2 million cells per mil is definitely optimal. But as we incorporate the enhancer into the production workflow, there's a really nice linear progression where we see significantly higher titers when we increase cell density to 4 million cells per mil. So when you're using the full VirusGEN LV kit, we definitely would kind of push you more toward those higher cell densities. And that takes us to our second polling question. As we mentioned before, there are many different hex cells in the world. Why did you choose your specific HEC293 cell type for LV and or AAV generation? And this is again a question where we want you to select all that apply. And so the options could include these cells generate the highest titers, regulatory support and GMP availability for the cell types that you're using, licensing or cost considerations. Maybe you've just always used them. It's what's always been in your lab. You haven't really thought about it. Maybe you're not sure why you use the HEC293 cells. And there are definitely groups that don't use HEC293 cells at all for virus production. Perhaps you're generating in baculovirus. So go ahead and select all that apply to you. And we'll give you a few seconds to answer this one. Okay, so that was a bit of a lead-in to this next slide here, where we're looking at transfection optimization per cell type. And this is looking at specifically adherent cell types for LV and AAV production. So AAV again on the top. And we're looking at 4 different unique HEC293 cell types. There's the 293 T17 cells, which do contain the large T antigen from ATCC. There's the 293 AAV cells from Agilent that do not contain the large T antigen. And then there's just plain HEC293s from ATCC and Sigma here, both again, not containing the large T antigen. And it's pretty clear that some cells do a much better job of expressing AAV than others. In particular, especially for those that don't want to use any HEC cell containing large T antigen, the 293 AAVs from Agilent are a really, really good choice for adherent cell expression. For LV, it's kind of a similar story. We see really robust functional titer production in both the T17s and the Agilent 293 AAV cells for lentivirus. And in this case, we do use the LV enhancer because it is compatible with adherent cultures, whereas the AAV enhancer is solely for suspension cultures. And again, we see that nice kind of 25%, maybe even twofold increase in titer when using the enhancer with both of the cell types. It's kind of hard to tell for the other two, the ATCC and Sigma cells here, just because they're so much lower in terms of expression level. So cell type definitely can matter. I think everybody in cell culture land has the HEC293 ATCC cells in culture or in their lab, in their cryotanks. But there are other ones out there worth exploring. All right, so one of the most critical aspects to look at for expression of both AAV and LV is looking at the reagent to DNA ratio and the total DNA amount used. In this graph, we're looking at AAV2 production in suspension cells, so using the viral production cells from thermal grown in XB293 expression media. And here we're looking at these 2 bars. It's 1.5 micrograms total DNA per mil at a 2-to-1 ratio of reagent to DNA versus 2 micrograms total DNA per mil in a 2-to-1 ratio. So if we're looking just at this, it's not a huge difference. We don't see a significant increase in functional titer when we move from 1.5 to 2 micrograms per mil. However, if we start looking much more closely at ratios, we see that we can double functional titer production when we find that perfect ratio of reagent to DNA for a specific vector set, cell type, media combination. All of these things do work in concert. So it's worth testing in your final cells and media with your final vector set, a series of ratios, really ranging from 1-to-1 to 2-to-1 to ensure that you found what is optimal for your specific workflow. And in this last figure, looking at optimization, we're looking at 3 different things. So there's total DNA, the reagent to DNA ratio, as well as harvest time. And for this, we're generating AAV2 in suspension cells, so the XB293F cells and XB media. The red bars are the 48-hour harvest, gray bars are 72-hour harvest. And again, we see that there is a nice uptick in expression when we move from 1.5 to 2 micrograms total DNA per mil of culture. So this is not per million cells, it is per mil of culture. So the cell seeding density was right around 2 million cells per mil here. So 2 micrograms per mil of culture. And we do again see that better performance at the lower ratio of 1.5 to 1 versus 2 to 1. And in the middle section here, we definitely see, that stronger performance when harvesting at 48 hours versus 72 hours. And so what we find with VirusGEN, as well as some of our other transfection reagents, because they do contain that lipid component, we do tend to see expression happen a little bit sooner, which may actually truncate your expression workflow and enable you to harvest at 48 rather than 72 hours. And in this next section, we want to look at VirusGEN for large-scale manufacturing, which essentially means how do you take a reagent like VirusGEN and then place it into a workflow that is like a 200-liter bioreactor? Is it compatible with that? And of course, we want to answer those questions for you right after you answer polling question number 3. So the question is, at what phase in your cell or gene therapy pipeline do you require a GMP-grade transfection reagent? In this case, we would like you to select just one. So is it process development, preclinical studies, phase 1 or phase 2 clinical trials, late phase clinical trials, commercialization, or again, there's the NA option for those that are not currently developing a therapeutic. So again, I'll give you a few seconds to answer that question. All right, so as I had mentioned before, when we were first thinking about VirusGEN and the formulation that we wanted to develop and how we wanted it to support customers, we knew that we would eventually need a GMP grade reagents. We started with research grade, I think, as everybody does. And so we do offer VirusGEN in 3 different unique phases, right? So there's the R&D, with RUO grade transfection reagent. The dispense sizes are smaller, again, to kind of fit those smaller scale workflows. So there's 0.3 up to 1.5 mil dispenses. And just today, we're launching a 30 mil research grade dispense as well for RUO VirusGEN. There's also the select grade, which is designed for, again, those preclinical and early phase clinical trials, larger dispenses, additional testing. So sterility, bacterial endotoxin, mycoplasma, and functional identity testing performed. Post-dispense, 30 mil dispense suitable for about a 10-liter culture, 150 mil dispense suitable for about a 50-liter culture. And then, of course, there's the VirusGEN GMP reagents, also with the same suite of quality release testing, 150 mil dispense, and fully GMP manufactured. Some of the questions that really come up for us when we're talking with teams that are in larger scale production, or they're thinking about larger scale production, we start hearing questions like, what is the complex stability? How long do I have in a complexation window? And this data is designed to show essentially that you have really anywhere from 15 up to 60 or maybe even 90 minutes for very stable complexes that can be formed at either room temperature or 4 degrees. But essentially, you're not stuck at just 15 minutes. You can extend that if you need the time to set up your larger scale workflows. We also found, as I had kind of indicated before, that VirusGEN is compatible with many different cell types and cell culture mediums. In this graph, we're looking at XP cells in XP293 media as kind of a benchmark, and then VP cells in LVMAX media, F17 media, XP media, all 3 from Thermo, and then there's the balanced CD HEC293 media on the far right here from Irvine. And we find that we see very robust performance in all of the medias. There does seem to be some synergy with the Irvine media, so it's one that we have been exploring more, and we also do encourage our customers to look at as well. So for those in large-scale production, another question that we get is, can we form complexes at 5% of the total culture volume instead of 10% of the total culture volume? If we're thinking of a 200-liter run, that's a 10-liter complex versus a 20-liter complex. And so we worked with a CDMO to develop this workflow where the reagent is diluted in complexation solution in one bag. In a different bag is the DNA. They're then combined, mixed very thoroughly. They can be incubated for 15 to 45 minutes and then added to cultures. And this works very, very nicely and does fit the time constraints for complexation, as well as the volume constraints for those large scale runs. And so the last data slide here shows functional versus genome copies per mil in terms of titers. And what we see, again, so either the 10th for the genome copies, either the 8th for functional titers, very normal here, but they do, in terms of trends, line up very, very nicely. So functional titers and genome copies, I think, are both really reliable methods for assessing your culture productivity. And here we see, again, that really robust performance from VirusGEN, even better performance for the VirusGEN AAV kit compared to two other leading GMP grade transfection reagents. And really, why have we spent so much time on this, right? Why do we think so much about improving titers? And really what it boils down to is that higher titers lead to fewer runs that are necessary to generate your therapeutic. So that's less hands-on time. It's fewer runs. It's a significant cost savings. And ultimately, the most important part is that it enables you to generate more therapeutic doses to save more lives. So to summarize our presentation, what we learned is that significant process improvements are really necessary to move from phase 1 to phase 2 of the cell and gene therapy kind of trajectory here to meet manufacturing demands and to make sure that we're keeping up with those larger assets production runs. And the VirusGEN GMP reagent and kits offer you that efficient DNA delivery for high titer AAV and LV generation. It is compatible with both adherent and suspension cells, so can be used really from the genesis of the concept of the therapeutic in R&D all the way through commercial manufacturing in whatever format you'd like to manufacture in. We do provide support for R&D from our R&D and application scientists so that when you do test the reagents, you can work side by side with a scientist that's very familiar with the process and can make sure that you find optimal within your own workflow. And of course, we're curious, so GMP compliant, no license required, completely synthetic and animal origin free. And the last thing that I do want to point out is that we do offer free kits for evaluation. So we definitely want you to test this in your lab to make sure that it does work well for you. And again, along with the free reagent, we provide you that support. So we'll work with you to make sure that you find the optimal ratios, you look at the right cell densities and medias as well. So that concludes the presentation portion, and I think we'll have time for a few questions. So I'll hand it back to Leah.
Okay, great, Leisha. So the first question is, do you think it's more important to optimize total DNA amount or reagent to DNA ratio?
Good question. So I actually see this question come up quite a bit when people are trying to decide what to test first, because you can only fit so many parameters into one kind of DOE style run. And I would say that looking at ratio is where I would start. So test those different ratios from 1:1 to 2:1, and then test total DNA amounts. But ultimately, you're going to want to look at both.
And do you have assays to show effective removal of enhancers through the purification process?
We do. We have partnered with a well-known CRO here in the US in developing what we call residual reagent assays. And these assays are, again, designed so customers can send their final samples to the CRO. They'll run the assay and ensure that there is no residual VirusGEN reagent or enhancers left in the final product.
And HcDNA is one of the major impurities that's challenging to minimize, especially for the higher dose of gene therapy. Do you have any data of HcDNA with your transfection kits?
That is a really good question. And at this point, we have not really delved into that question. It's something that we definitely want to look at in the future, because I agree it's a key thing to understand and assess.
And does your transfection reagent promote the excretion of AAV particles into cell culture medium so the cell lysis step can be avoided during the harvest?
Another really good question. So I would say that that would be handy, but it's so much more serotype dependent than expression reagent dependent. So certain serotypes do tend to leach more into the culture media. Some are maintained within the cell, and our reagent versus other reagents really won't change that. Ultimately, what we see, even with serotypes like AAV2, doing the whole culture lysis is what results in the highest titers.
Okay, so it looks like we have time for just one more question. If we didn't get to your question, we will be passing those along to Leisha, and she can follow up with you directly. So go ahead and continue to type your questions in. But the last question for the webcast is, how much reagent and enhancer do you provide in an evaluation kit?
Excellent. Yes, so our evaluation kits are designed to be generous because we want you to be able to test different parameters, including the ratios, the cell densities, total DNA amounts. So within each evaluation kit, you'll receive material sufficient for about 1 liter of transfection. And so I know most groups test in about a 30 ml format. So that really does enable quite a bit of testing to make sure that you're comfortable with the reagents and can find parameters that are suitable for your workflow. So enough for 1 liter worth of transfection.
Okay, so thank you to Leisha, and thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you. Bye.